Hello everyone, and welcome to Breeding and Genetics webinar. My name is Lucia Barreiro and I am the editor of Hatchery Feed Management. This is the second webinar in the series Trends in Aquaculture Hatcheries that we are holding, focused on different topics of importance for hatcheries such as diseases, breeding and genetics, and feeds. One of the keys of the growth of an economically and environmentally sustainable aquaculture industry is the genetic improvement of the species that we grow. In the species where these techniques have been implemented, such as salmon, tilapia, and shrimp, farming efficiencies have improved, increasing farmers' profitability. For today's sessions, we have a great panel with major players that will discuss some of these examples and their future perspectives. Thanks to the support of our sponsors, Bechmark Genetics and the Center for Aquaculture Technologies, this webinar is open and free. Each speaker will give a 15-minute presentation, and then we will get to a final panel session to answer questions from the audience. To submit a question, please type it in the Q&A box, and all webinars in this series will be available on our website and YouTube. I want to especially thank our assistant editor and webinar manager, Marisa Yanaga, who's making sure all our webinars work. And finally, it's my pleasure to introduce Anna Sonesson, who will be moderating this webinar. Anna is Research Director, Breeding and Genetics at Nofima. She is a researcher on genomics and selective breeding, including genomic selection and QTL mapping analysis. Anna also works on methods to control inbreeding and inbreeding schemes and on the design of aquaculture breeding schemes. She works on a number of species, including Atlantic salmon, European sea bass, microalgae, and plants. Anna, hand it over to you.
Thank you so much for inviting me and for this nice introduction. Yes, so today's agenda is that we will first have the 4 presentations, and then there will be no questions after each of the presentations, but they will all be coming on board when we have the panel at the end. So now we just start with the first presentation. That is by John Buchanan. He is the CEO at the Center for Aquaculture Technologies. He's the founder of this institute, where he leads a team of professionals dedicated to the application and commercialization of innovative technologies for use in aquaculture. John is a pioneer in the application of genome editing to aquaculture and has led research teams focused on the use of traditional and advanced selective breeding techniques, hatchery technologies, and genomics for improved productivity in shellfish, crustaceans, and finfish. Before the current position, John served for 5 years as the Director of Research and Development at Aquaculture Technologies and has held research leadership positions at Louisiana State University, University of California, San Diego School of Medicine, and Kent Seatech Corporation. The title of his talk is Future Perspectives on Selective Breeding in Aquaculture. The floor or the screen is yours, John.
All right, thanks, Anna. Let me share the screen here. Did it come through okay, Anna? Okay, thanks everyone. So I'm speaking today from California in the United States. It's very, very early here, with 5 o'clock in the morning or something crazy like that. So I will try to be awake and attentive as a speaker, but luckily there's questions at the very, very end, so I can wake up a little bit still. So we're, as Anna said, thanks for the introduction. We're the Center for Aquaculture Technologies. We operate facilities on the East and West Coast of North America and Canada and the US. We're a contract research facility, so we support other companies in the industry by providing services, doing health challenge trials and diagnostics, nutrition trials and feed production, supporting research and development, breeding and genomics, and genotyping. And of course, if you're ever in the area post-pandemic, please stop by and say hello. We'd be happy to give you a tour. So selective breeding is in general the concept of increasing genetic performance in your animals over a period of time. And so each generation you gain advantages, and there are many applications of this, ranging from disease resistance to growth and other obvious traits. to some more interesting things, reaction to feeds, the different production systems you can use, environmental consequences and considerations, and sustainability. So it's a really dynamic and active field. The most famous success so far has been in Atlantic salmon, and this was work in Norway started in the 70s. Over the first 5 generations of domestication of the wild broodstock, you can get profound gains in performance. You know, in this case, they've shown over a doubling of the growth rate, and importantly, an improvement of 23% in feed conversion by applying basic selection and selecting for improved performance. So the generation time is 4 years, so 5 generations takes quite a long time in salmon. In other species such as shrimp, we have some advantages, and the generation times are quite short. So both are important considerations. When you look at the applications of breeding, there's this continuum from very simple applications such as making hybrids or crossing different strains or understanding what strain you're working with, through simple projects like mass selection, picking the best looking animals and having those become the next generation of broodstock, using pedigrees for either within or between family selection or combined family selection. So now you can map out the relationships and use quantitative genetics to more accurately predict the next generation of broodstock with improved performance. And then getting into the area of using genetic and genomic markers to help identify traits of that importance. And then ultimately, at the most sophisticated level now, we can use genomic selection. So using genomic prediction to improve the accuracy of selecting the next generation of broodstock. And in general, as you go up this arrow, you get increased genetic gain per generation, but there's also increased genetic complexity in applying the technologies and increasing cost. So there's a trade-off in the best strategy, and it can vary greatly based on the size of the breeding program. For small, simple programs, mass selection may be the best choice, but for larger, more comprehensive programs, the world is moving toward genomic selection. And overall, for genetic gain, the main characteristics are heritability, and I should not neglect mentioning the importance of collecting good phenotypes, so being able to measure the animals well. And then the intensity and accuracy of selection are very important, and that's where these genomic tools can really be of benefit. So as we're looking to the future is moving up this arrow. And the way we can use genomic tools is we look at DNA variants. So these are changes in the DNA, the basic coding within an organism, and there's differences between individuals. So sometimes these variants are a single nucleotide variant. So there's one base pair change between an individual on the top line, that's the DNA for one individual, and a second individual on the second line there. But there are other types of variants as well. So you can have deletions, so some DNA is missing from a relative, or there's substitutions, inversions, or other larger changes. So this variation forms the foundation for selective breeding programs. And there's larger structural variations as well. So if you look at a gene structure here, you can have many different things: insertions of DNA, deletions that occur, relocations of the different parts of a gene, or other pieces of DNA. And these affect performance. And selective breeding programs, whether they're mass selection or sophisticated genomic tools, are looking for these variations that occur in DNA and finding the ones that lead to better performance and traits. An example of this is if you take the wild mustard plant, domesticated in Europe and the Mediterranean region 3,000 years ago, selection for different parts of this one plant over time have led to these species of vegetables, from kohlrabi to kale, broccoli, Brussels sprouts, cabbage, cauliflower. They all came from the same foundational plant, selecting for different variants in DNA over time, selecting for the fruit or the leaves, led to significantly different vegetables. And we've all seen other examples of this in our favorite friends. In dogs, of course, there's huge variations in breeds that have been accrued over time. No genomic tools, but just selecting inherently for variation in DNA. So as we get more sophisticated in how we apply breeding, we begin looking at DNA markers. So instead of just looking at the phenotype, you can begin to look at the genotype. And for this, we use SNPs, so single nucleotide polymorphism. So these are those one base pair changes. That's a marker we can easily measure in the DNA. And we also have genomic tools becoming available. The shrimp genome was recently sequenced, and there's 1.6 billion letters. So basically these stretches of DNA, 26,000 genes. There's 7 million of these SNPs that you can find. And we've moved towards SNPs as the marker of choice because they're numerous. And even if they don't cause a change, they're frequently linked to a change. So we can use to predict different changes in DNA that we want to select in a breeding program. So these SNP markers allow detection of variants, and these changes in DNA function can form the basis of selective breeding. And so if we're thinking of a stretch of DNA here, and we're going to look at SNPs, the SNPs are each where there's a SNP, you can say there's a tiny lamp that's there. And as we move through and use these tools, they illuminate the surrounding DNA. And over time, they're defined within an individual, and they give us information. So if we can genotype or look at the SNPs in an individual, we can gain this information, and then you can begin to compare individuals. So they'll have they'll be 99 or more % identical, but there are these small changes in these SNPs that we can measure. This allows us to do interesting things. If you look at population of individuals and genotype multiple individuals, and they all have very similar SNP patterns or genotypes, you can conclude there's very low diversity in your population, and inbreeding is a problem. Conversely, if you see a large variation in the SNPs, you can begin to think, well, we have good diversity, there's low levels of inbreeding. So even if you're not doing really massive selection projects, you can still get information on the health of your population and how you might go forward if you have a mass selection breeding program, for example. You can do more sophisticated things with statistical calculations and begin to look at structure in populations using these SNPs or low density SNPs. And this can be done with only 100 to 200 SNPs. And you can look at and say, if I have multiple lines in my population, are they really different true lines? So on the top here, the farmer was maintaining 14 different lines of shrimp, and in the end, they really only had 3 populations. And then on the bottom example, there's a farmer that had 10 lines of shrimp. All were genetically distinct and well maintained. So this can be important in managing simple breeding programs as well as more complex breeding programs. And then finally, you can use these 100 or 200 SNPs to assign progeny to parents, which is very important in family-based breeding programs. And so you can look and see the combination of SNPs that were in the parents and look for those in the progeny. And in that way, without tagging or keeping family tanks or investing in a lot of complex capital, you can genotype and reestablish your pedigree and relationships and allow for efficient selection. So this is more or less state of the art at the moment in many breeding programs in different species. Of course, there's a wide variety of species in aquaculture. If you go forward and become more sophisticated, we have these low density SNPs. So we have 192 SNPs. But if we're looking to the future is having more markers available. So you have more of these lampposts that are available in the DNA, which illuminates more of the genome. And at this point, we are capturing enough information from the genome to do some interesting applications beyond pedigree assignment and looking at diversity. So for example, we can look for SNPs that are linked to traits. So if you take a population of animals, you collect phenotypes, and you can genotype them against these 50,000 SNPs. The ability is to find SNPs that are linked to the trait of interest. And so you can have a statistical association, find a subset of SNPs that you can use in a breeding program. So for example, if you find SNPs that are associated with disease resistance, you can use that and genotype your broodstock, identify broodstock that have those markers, which then predicts that they have the trait you're interested in. And you can use those, and the progeny will have improved genetics compared to the previous generation. This is a very powerful tool called marker-assisted selection. In addition, you can do things like, and it's a study we ran a few years ago, we're looking in response to feeds and salmon. We can produce high soybean-based diets and find markers or SNPs linked to performance of the fish on the high soybean diet. So rather than modifying the diets for better performance in fish, you can also select for the fish to have better performance on more economical or environmentally friendly diets. This is very interesting and powerful tools that we can use. And then ultimately, if we have this information of the SNPs and the markers throughout the genome, as you're looking to what's the future will hold, genomic selection is the next step. And this has been well proven in Atlantic salmon as a valuable tool. And it's moving into other species, for example, shrimp and tilapia. the basic premise here is that you have a population of animals, and you can take a subset of these animals and collect a phenotype on them. In this case, we have large and small shrimp. And you have your parental population, that's a separate group. But if you collect genomic data from these animals, and you know the performance characteristics and the genome and the marker scores, you can look back in the siblings that are in a secure breeding facility, for example, and find those that have the same genomic information. And those will become the next generation of broodstock. And this, again, increases the accuracy of selection, which increases the genetic gain per information, because we're not looking just at the phenotype, we're looking at the whole genome of the animal. And in practice, you can imagine scenarios where you have a secure breeding nucleus where you're controlling for disease, and you have your broodstock there, you produce fingerlings. Some fingerlings leave the nucleus for disease testing or other phenotype characterization. For example, early maturation is a problem in many finfish species. The breeding candidates remain in the facility, so you can collect phenotypes, genotype the animals, and the genomic information flows back into the breeding nucleus. So this is where we're headed, I think. And as we control disease in our breeding populations, we can still collect phenotypic information, move it back into the breeding nucleus as genomic information. And the more animals that we test for phenotypes, that increases our accuracy. And the more that we genotype, that increases the intensity of selection. So you can really make a big impact on genetic gain in these situations. If you look even further into the future, so we have these high density SNPs with 50,000 SNPs available that cover a lot of the information of the genome, so we can see the genome well. Where we're going next is looking at whole genome sequencing. And this is the ability, instead of 50,000 SNPs, you can look at 2 million SNPs. So this is a giant number of SNPs. It's not that much more coverage than you might get from several thousand SNPs, but it is fundamentally deeper coverage. And so you can begin to look with even better accuracy for traits. And the way this is looking, we're working on technologies to make whole genome sequencing roughly cost equivalent to the approaches we take now for looking at 50,000 SNPs. But most importantly, as we look at the whole genome and look at this really deep coverage, we can begin to see the causative variants. So not just SNPs that give us linkage so that we can see where other traits are and make predictions, but the actual fundamental changes in the DNA that are causing the trait. And at that point, we aren't predicting, we are detecting directly in the DNA. So this is where we're moving, sequencing the whole genome of your candidate broodstock, understanding which ones have the causative variants that have occurred naturally that you want to select for, and having those become the next generation of broodstock. So we're still proving this technology, but it's been implemented in cattle, and we've done some work as well as others. So this is really where I think breeding will be moving in the future. And as we understand the causative variants, and there's whole fields of genomics beyond this that are devoted to understanding what's causing differences in phenotypes at the genetic level or at the gene level. The next step really, as I've said, going into the future multiple times here, but really going into the future is the potential of genome editing. And so on this continuum that we talked about in the beginning, genome editing is at the top. It's the most expensive and most complex to execute at the moment, but also gives you the opportunity for significant genetic gain. So if you remember back to our fish that's hopping up the ladder of genetic improvement over time, genome editing gives us the option for certain traits to make a giant gain in genetic performance over one generation, using understanding the causative variants that we want to find and then creating them in the animal. So it's not anything different than you would do in a selective breeding program, looking at thousands or millions of animals for the variants that you want to improve performance. In this case, if we know the variant, we can create that variant in the animal. And so these happen, random DNA variants occur in fish and shellfish and crustaceans naturally. So there's all these breaks and rearrangements that we talked about before. So this technology, as I'm sure everyone's heard about at this point, uses different types of enzymes to make these breaks. So CRISPR is a famous example. There are talons and zinc finger nucleases. there are ways to create these in a very precise and directed manner so that you can create the variant that you're trying to select for just all in one time. And the way this is done, instead of selecting for the animals through a large number of individuals, this is a picture from our lab where we're injecting shrimp eggs. You can take the enzyme and a piece of RNA that directs the enzyme specifically to a location in the DNA, inject it into a newly fertilized egg, and create the variant that you're looking for. There's no new DNA added. It's not transgenic, but it is a way to supercharge the selective breeding process. And the applications are fundamentally the same as for selective breeding. There's many things you can work on, including sterility. So one of a big focus for us is solving the problem that we have in aquaculture of early maturation during production, and also environmental concerns with farmed fish because they're so closely related to wild fish. So if we can find ways to have sterile fish to go out into the production, and we're using genome editing for that, that's a big benefit to the industry overall, both from the environmental and sustainability perspective and increased performance. There are other applications for which we need rapid improvement. So for example, resistance to disease or growth rate, you could increase flesh quality, feed conversion efficiency, and waste output. So really the same as we talked about before, but you can increase productivity, increase nutritional benefit, and decrease environmental footprint using genome editing, just as you could with selective breeding. And interestingly, the world seems to be trending toward acceptance here. So if you look at the current guidance for regulation of genome editing, and this is a figure taken from the Genetic Literacy Project website, which if you're interested in this, I encourage you to visit the website. They do a great job of explaining in a very neutral way the technology and the regulatory environment and lots of other good information. There are 3 countries, Brazil, Argentina, and Paraguay, who have stated unequivocally, if there's no new DNA added, you can use genome editing just as you would any other selection technique. And then in addition, Japan, Australia, and Canada have recently indicated that their regulations will require notification to the government. So you have to let the government know what you're planning. But again, if there's no new DNA added, genome editing will not be regulated as a technology, which is a really good step forward in our goal to improve genetic performance of the fish and ultimately feed the world. Central America, the US, and the UK are in conversations. In those cases, as you can see on the left side of the graph here, in crops, there's been much greater acceptance of this technology. Animals are lagging behind, but 3 years ago there was nothing green on this figure, and now we have some countries who are open to this technology. And I think the rest of the world, notwithstanding Europe, which is always quite difficult, but the rest of the world seems open to this idea that we can use this technology as another tool in the breeding toolbox to increase performance. So in the end, if you think back, the first written documents on fish culture were from China in 475 BC. That's when they first started selecting carp. And we've been modifying fish genes by selecting variants ever since. And the toolbox that we have ranges from mass selection through pedigree-based selection, using marker-assisted selection, and ultimately sophisticated genomic selection, and then genome editing as resources that we have for improving. And as we go down that list, the genomic resources become a larger consideration and the focus of a lot of research and development work and a lot of really fantastic work in statistics and modeling. The important thing here is these are all just tools in the toolbox. In any solution, any circumstance, there may be different applications that are most appropriate. So cost-effective solutions are essential. You can't go in, without, especially in aquaculture, where we have many broodstock, and in contrast to cattle, for example, the individual broodstock is not as expensive. A lot of the focus of our company and many of other researchers are how to find solutions here that are cost-effective and we can apply to the industry. And they're all just tools in the toolbox. In some cases, pedigree-based selection is going to be the best approach. In other cases, pedigree-based selection combined with genome editing may give you the best strategic approach. But it's really an exciting time in the industry, and a lot of things are happening. And I think will be a lot of change occurring. And there's a final note at the bottom of the figure here is the word phenotypes. Probably the most important part, in spite of all this talking of great genomics and genomic selection, is the ability to collect and reproducible phenotypes and do a great job there. And that's the key that underlies everything. But otherwise, there's lots of tools available and the future looks very, very bright. With that, I'll say thank you very much. Back over to you, Anna.
Thank you very much, John, for this nice presentation. I think we just go over to our second speaker. That's Dr. Marcela Salazar. She works as a scientific director at Bechmark Genetics, Colombia. She has been studying genetics of immune response against viral and bacterial infections for more than 20 years. From year 2000 to 2016, she was scientific director of the Center for Aquaculture Research in Colombia at CENIAQUA, working on the immune response of Vanamay shrimp against virus using white spot disease as a model and in the selective breeding of Vanamay. In 2016, she joined Bechmark as the scientific director of Bechmark Genetics Colombia. She has published several articles in peer-reviewed journals ranging from Vanamay to human diseases and immune genetics. Her title is Breeding for Disease Resistance in Shrimp, from Mass Selection to Genomic Tools. Please, Marcela.
Thank you, Anna, and thanks for inviting me. So let me share my screen. This is a talk that is going to be really easy to do now with the pandemic, because in the past year, everybody has been an expert in all the terms that we use in epidemiology, pandemics, herd immunity, variants, everything is common knowledge now. So that makes my talk very easy. And also thanks to John, because you explain all the genomic selection, so I don't have to do that again. What I'm going to show you is basically what John showed, but in the real world, how we have been applying all the technologies in shrimp. First, I would like to start my talk explaining what's the difference between tolerance. Probably some of you have heard that again time and time, but I still find that there is a confusion on what is a resistance and what is tolerance. Resistance is the ability to limit the pathogen load, the ability to prevent an infection, to combat the pathogen, while tolerance is the ability to deal with the given pathogen load. It's how to live with the pathogen. We don't fight it, we live with it. So there are two complete different ways to respond to a pathogen and two complete different ways to select for in breeding and genetics. In the world of shrimps, we have had many pandemics, at least two in the last 20 years, white spot and apins. We have had many epidemics, HHMV, INMV, NHP, TSV, and we have minor diseases. So it's a word where we have been using the word pandemic since many years ago. And we have a problem. We don't have a lot of tools in the world of shrimps. We don't have vaccines. We are all waiting for a vaccine to change the world to the normal again. In the shrimp world, we don't have that. We don't have therapeutics. It's very difficult to get a drug that works in shrimp, how to administer that, how to treat them. But we have surveillance, we have management, and in management, we use decreased densities, for example, which is the equivalent to trying to keep distance. And we have a tool that we don't have in humans, which is selective breeding. We can try to select animals to be more resistant to diseases. As John said, we can go from mass selection to genomic selection. We have gene editing, although it is not allowed yet, but it's a tool that we can play with. The first tool that we have in the genetic resistance, because I'm going to talk about breeding, it's mass selection, which has been used since the beginning of aquaculture in the world. And it is only to get the best animals in the pond. In terms of breeding for disease resistance, what we do is we submit animals either to a control challenge test in lab conditions or in a pond with an infection, and we just take the survivors and reproduce them. That is pretty easy, and it works in some of the diseases. Then we go the next step that John explained, which is okay, if that works, let's try to do a family-based selection and using SIP-tested traits to try to be more biosecure and not use infected animals. In this one, you just get a lot of families, 40, 50, 60, 100 families, get a subset of animals, test them for growth, test them for other traits. In this case, this is challenge. You just send a subset of animals, challenge them, and see which are the best families that survive and take siblings for those families. It's very effective for disease and flesh quality traits, but it's not as effective as within family because we are losing the between family component. So we'll get only 50% of the variance. It can be combined then with using genomic selection, QTL, and all of other tools that John just explained. The next step, logical, is okay, if I know that we have very good families, let's try to see what are the genes behind those families. If I can find the variant, if I can find the marker. When we talk, John was talking about linkage disequilibrium. If you have a panel of 50K, as you were showing, or 192K, or 2 million K, which you would love to have, then you will probably be able to find either the variant or a marker that is really close to the variant. And this is what is called linkage disequilibrium. The way I explain it is when we went to the university, for example, we always had very good friends. And if you see one of those, probably I was with him. So the idea of linkage disequilibrium with the marker is if you see that marker, probably you will have the gene or the SNP that is responsible for the trait, in that case, resistance. So QTLs are controlled by many genes, which differ in size and effect. So we can identify the genes. If we are lucky, we can identify a QTL that explains a lot of the variance, 20, 30%, and it's like hitting the jackpot. If not, we'll have to go to gene-assisted selection and genomic selection. In genomic selection, we try to look at the most of the genome of the animal that we can, to see if we can small pieces that will give us small changes that will give us an increase and then be able to select more accurate. In this time, we are looking at trying to look at the whole genome to see which animals should we select based on the genes. It picks up the QTL effects on all SNPs across the genome, and it's more efficient and more accurate as the number of SNPs increase. So we'll be able to be looking at the whole genome in very fine detail. Holsteins are the genomic selection pass for cows. This is a very nice paper explaining how that works, especially in Holstein, because they have been doing breeding for many years in traits such as milk production, protein, fat, etc., etc. So if we can get increase in that species that have been selected for many, many years, we have a lot of expectancy and a lot of possibility to get gains in shrimp or in fishes. This is to show you if we do the genomic selection, if we only do the family breeding, we'll be looking only a small number of families that have the best individuals, because we're afraid of looking at another animals that could be as good as the others, but we don't know the information. When we do genomic selection or when we do SNPs, we'll be able to select animals from the families that are not as good as the others, but animals that have the same genomic marker or the same genomic appearance that the others, and we can be sure that they should work. So we'll have an increase in our selection differentiation, and we also will be able to decrease in breeding because we'll be taking animals from a lot of families instead of only from a few families that we would do with the normal breeding values based on only family selection. So in terms of selection accuracy, if we go from family selection to genomic selection to mass selection, the best for disease resistance would be to be able to do mass selection, because we'll be selecting the animals that really survive and also animals that have an immune priming effect. In the past years, there's been a change since when I started in the shrimp world. Everybody said, oh, shrimps, they don't have immune system. I was like, everybody has an immune system. Every animal, plant, they have an immune system. It's not the same that we have, but they have one. Then they said, oh, they only have an innate system. They don't have memory. And now the tune is changing again. They have sort of a memory, and it's called immune priming effect. It means that if we prime the animal, it will respond better the next time. Or even if we prime the parents, they will respond better for the sequel, in the F1. So this is in terms of selection accuracy. But what happens when we talk about biosecurity? Then mass selection is not a biosecure, because we'll be infecting the animals and they can be transmitting the disease to a progeny or infecting other animals. So the best would be to go to family and genomic. Now I'm going to talk in the real world what we do in Colombia. Colombia is a country with 2 coasts, the only country in South America. I always like to teach a little bit of geography. And we have a breeding program in Cartagena, and we have a breeding program in Tumaco, one in the Atlantic coast and the other one in the Pacific coast. Why do we have 2 programs? Well, that goes to the history of diseases in Colombia. The first that I'm going to talk about is Torah syndrome virus, which came to Colombia in 1994. At that time, it killed 70% of the animals in all the ponds. And immediately, the sector started using pond survivors as breeders, and they increased survival by 30% in 1 generation. So this is the first example of mass selection in ponds that works perfectly. With that result, Colombia decided to start a family selection in 1997 with the help of AFGC at that time. In a typical challenge test for Torah, it was very nice. We had families from 90% survival to families from 30% survival. So very easy to select the best families. And from that, to use the siblings as breeders for the second step in the breeding program. Torah was a success, and it completely disappeared from Colombia and from the rest of the world, actually, because most of the populations are resistant. The heritability of that disease is between 0.20 and 0.30, so it's a perfect virus to play with. Now we have another one, which was white spot. When white spot hit Colombia in 1989, between 2000 and 2004, it didn't work. I'm going to show you why. And then we had to go back to mass selection from 2008. Why it didn't work? Because we had a negative correlation with growth, very marked, ranging from -0.5 to -0.20, meaning that the best families for growth were the worst families for white spot survival, and vice versa. And in the challenge test, we had only a couple of families that survived about 10%, and the rest of the families didn't survive. So after a while, trying for 4 or 5 generations of doing genetic selection, of doing family selection, with only the heritability of 0.04, 0.05, we decided to change strategy. Before doing that, we did a lot of analysis on a differential gene expression, finding crusting and C-type lectin in the survivors. We analyzed the effect of apoptosis on white spot. We identified the effect of hyperthermia. That would be really nice if we could treat, for example, SARS-CoV-2 with the same heating or with a fever. And after that, we decided to do a change of a strategy and go back to mass selection to see if we could really find some survivors that we could play with. So we infected millions of animals at PL stage, PL40, including a lot of populations. And in the first challenge test, we had survival of only 1 in 100,000 animals. So when we reproduced those only survivors that we even had a name for them, we found that the progeny was very good. It had a 50% survival in a challenge test, where another population, the untreated population, which was the Cartagena line that was non-selected for white spot, had a 2 or 3% survival. We did that in challenge test in the University of Arizona and also in Colombia, showing that. So now we had a population that were resistant or very good for white spot, but was mass selected, and we had our population in Cartagena. So we said, okay, what happens if we cross both? If we do a hybrid, an F1 of the Cartagena and the Tumaco population? And it seems like we were lying when we got the results. It was 6% in the susceptible, 26% in the hybrid survival, and 54% in the resistant, in the tomaco pure line. So with those tools, we decided to play with and try to combine the breeding nucleus for resistance and use that to identify with the genetic tools that we had and with genomic tools, try to develop an SPR from SPF population. So what we did was take a parent line, for example, pure tomaco and a hybrid with Cartagena. We did a training population. What is a training population? Is a population that we infect in that case, or that we use for our phenotype recording. In that case was a white spot challenge. We challenged the training population and selected all animals, recorded the time of death, the day of death, and did a genotyping with a 35K SNP chip. And we also kept our breeding candidates, which are the siblings of the same animals. When we got the results from the genotyping and from the challenge test, from the phenotype and from the genotype, we decided to select animals based on those performance for reproduction. And what we did was do a genomic selection proof. Okay, our question is this working? Is the genomic values that we are giving associated with good performance of the animals? When we did our GWAS results that John showed also, we couldn't find any interesting or a very QTL trait, but we found a lot of QTLs explaining a small portion of the genetic variance. So with this, as I told you, we decided to do 3 populations to see how it was working. We did a good EVV, meaning we selected the animals that we believed based on the genomic breeding values that were very good for white spot. We selected some that were very bad for white spot. And we had a random, which was not completely random since we had taken the high and low animals, but we selected the 3 populations, did random mating within the group, avoiding full or half-seed meetings based on the genomic relationships. And we produced 60 families, 32 good, 20 random, and 8 bad. And we re-challenged them under control conditions and did all the analysis again. In that final challenge test, we found that our high or families that were selected based on the high GVV had a 53% survival, while the ones from the low GVV had only 26% survival, getting a genetic gain of 14% in only 1 generation of using only genomic selection. In that, we hadn't challenge test the parents. They were only the siblings based on the genomic selection. As John showed, we have become a lot of way from the wild animals, domestication, then selective breeding, then optimal selection of production traits, then the new technology methods. Then now we are going to CRISPR if they allowed us. And what we are working now is on going to single nucleus sequencing to try to identify which are the genes within each cell that is infected or not infected in different tissues that are responsible for the survival in those animals, or what is changing. So this is what we are doing now. And it's very interesting, as I'm telling you, to talk about pandemic in this work, because in shrimps, we have these technologies that can make for the lack of vaccines or therapeutics. We have been using technology transfer from the world of salmon that is really, well, they've been working for a couple of decades on genomic selection. And we are transferring that to shrimp, tilapia, and other farm species, or such as sea bass and sea bream. Thank you very much. This work was performed also with the cooperation of Nofima, Nick Robinson. We had very good time working with him in the past 3 years, and from all the team in Colombia. Thank you very much.
Thank you very much, Marcela. We will run off to the third presentation. It's by Rajesh Joshi. He's a senior researcher at GenoMar Genetics. Rajesh has a background in veterinary medicine. He's senior researcher at GenoMar Genetics and is responsible for planning and implementing selective breeding in 3 breeding nucleus of night tilapia in the Philippines and in Brazil. He's also a visiting associate professor at the Himalayan College of Agriculture Sciences and Technology at Purbanchal University in Nepal. His title is Past, Current and Future Trends in Selective Breeding of Night Tilapia for Production Efficiency. Please, Rajesh.
Yes, thank you, Anna. Let me share my screen first. Yes, thank you for the presentation. Thank you for the introduction. So I will be presenting about past, current, and future trends in selective breeding of Nile tilapia for production efficiency. Today, I have divided my presentation into 3 different parts. First, I will briefly describe who we are. And then in the later phase, I will describe about global trend in Nile tilapia production or tilapia production based on production volume, technology, and threats, and where do we stand now as a company, and finally, what does this mean to the farmers?
Can I interrupt? Can you? We don't see your screen. Can you share?
You can't see my screen.
No.
No.
Okay, I'm sorry. Just a minute. Share. Can you see it now?
Yes.
Yeah.
Thank you.
Okay. You see my full screen or half screen now?
You need to put it in presentation mode. There you go.
Now it is fine.
Good.
Yes.
This is good.
Okay. Yes, thank you. Sorry for some technical problems. Yes, we are a part of a large group of primary breeders. We are a part of EW Group, where the EW Group is responsible for breeding in multiple species like layers, broilers, turkeys, and fish. And I hope if you know about other species, then you recognize the breeds or strains on here. In the fish, we have a sister company, AquaGen, who is responsible for salmon and then trout breeding. And then we GenoMar are involved in Nile tilapia breeding. The advantage of having these different groups of primary breeders is we can have a very fast technology transfer and very good implementation of new technologies across the species. Our breeding nucleus, as Anna mentioned, that we have 3 breeding nucleus. So we have 1 breeding nucleus in the Philippines, where this breeding nucleus is for the GenoMar strain, and we have other 2 breeding nucleuses in the Brazil. In the Brazil, we have the breeding nucleus for 2 strains or 2 brands, Aquabel and Aquamerica, and we have our R&D. R&D and genetic operation is in Norway. The R&D operation, we are located in the Norwegian University of Life Sciences, where we plan and implement the breeding programs in these countries. Our distribution operations. We GenoMar Genetics are the largest producer of tilapia seeds in the world, with the capacity of around 500 to 600 million fingerlings and 50 million juveniles. In total, we have 60 hectares of fingerling production infrastructure. And currently, there are around 250,000 females to collect more than 1 billion eggs. And we are present in 5 different countries, as can be seen in the map. In the Asia, our distribution operation is present in Philippines, Vietnam, and Malaysia. Whereas in Latin America, our distribution operation is present in Colombia and Brazil. And in Brazil, we have different sites to provide the fingerlings to the market. Why genetics, or the power of genetics? Previous presenters, John, Marcela, they have described very beautifully the advantages of different phases of genetics. I just want to summarize it with these 3 figures. The first one with the maize. The A is the wild variant, and D is the present farming variant, where it has gone through a lot of steps in domestication and selection. In the middle there, it is an example of the Atlantic salmon for the disease IPN. In the y-axis in the left-hand side, it's number of disease outbreaks, and in the x-axis is the number of year. As we can see that number of disease outbreaks in IPN, when the vaccine was available, it was around in the range of 180 to 220. And in around 2009, AquaGen, they introduced the genetic IPN resistance strains. And after that, the incidence of IPN has been decreased a lot. So this is the power of the selective breeding for this is one of the great example for selective breeding for disease resistance. And everyone knows the example of poultry, where they have been selected for growth and other traits. And this is in 56 days, if the strain or if the poultry used to weigh 905 grams, currently it weighs at around 4.2 kilo, which is the function of both improving the genetics and then the nutrition. And it is expected that this is around 80 to 90% the genetics. The tilapia value chain, if we divide the tilapia value chain, we can divide it into the breeding nucleus, multiplier activities, hatchery activities, nursery, and the final producer farmers. We as a company, we have divided it into the genetics and distribution part. The first is the R&D or the breeding nucleus, where majority of the R&D activities takes place and gets implemented. And from the breeding nucleus, around 20 animals are selected as the great grandparents. And these go through the multiplication phases, systematic multiplication phases to produce the parent stocks, fry or fingerlings. These parent stocks are then sent to hatcheries, where they are sexually matured, and then they produce fries or fingerlings. As we can see that the number of animals in different stages increases. And then from the hatcheries, the fingerlings or juveniles are sold to the farmers, where they are growed out, harvest processed, and the fillet or the whole tilapia is sent to market. So this is briefly about tilapia value chain. And where are we involved in this? We are involved in the genetic improvement. That is, we improve the biological, economical, and environmental performance of tilapia aquaculture. We document the genetic progress and maintain the genetic diversity in our populations. Whereas in dissemination, we make sure that we have the year-round availability of the seeds or fingerlings with cost-efficient and sustainable technology for seed production and freedom for diseases. If we see the trends in global production of Nile tilapia, this has been described in the paper by Kumar Ganet and Carol R. Engel in 2016. This growth phase can be divided into 4 different parts: early tilapia farming, and then initial growth phase, early growth phase, and rapid growth phase. And we can see that the production of tilapia is increasing every year in different phases. In the early tilapia farming, there was a limited and no control over reproduction, and then the production technology was extensive in the ponds with no any controls, so the production was low. Whereas after 1980s, it was possible to control the reproduction, and then at that time there was hybrid production technology for tilapia was introduced, and then the extensive pond cultures started, and we saw that the production, global tilapia production, was increasing. Then after 1990, it was possible to sex reverse tilapia fingerlings, because as we all know that the males grow faster than females in the tilapia. And one of the, I think, the great was the establishment of gift and other genetic selection program. And from the extensive pond cultures, the aquaculture system was semi-intensive and intensive production systems, and then various nutritional studies happened. And after 2000, now we are in the rapid growth phase, and I think tilapia is one of the important freshwater species now. After 2000, it happened, this rapid growth phase is due to the rapid dissemination of the gift strains to various parts of the world, and then other genetic selection programs and improved strains were also established. And there has been a rapid technological advancement in the Nile tilapia breeding and selection programs. So let me just give an example of how we, GenoMar Supreme Tilapia, one of our strains, went through all this phase with more than 30 years of R&D and breeding work in Nile tilapia. So in initial phase, this goes back to the ICLARM in 1987, where they brought the 8 different species from different parts of the world, majorly from Africa, in the Philippines, and did the cross mating between 8 different strains and produced the gift strain. And then in generation 4, or in 1991, they started to select for the growth or low average body weight, that is around 200-300 grams of fish. And in generation 10, 1999, GenoMar bought the commercial GenoMar, then bought the families and started in the commercial farming commercially. Then in the rapid growth phase, with the advancement of the technologies, we started to use the microsatellite based parentage assignment for this our selective breeding. In in 2006, we started to select for higher average body weight. That is the fish around 800 grams to 1 kilo. And then we started to select for the fillet yield also. Then in 2010, we started to select for robustness. In 2017, we were brought by the EW group, and then we also bought the AquaBel strain. And then here, we started selecting for Streptococcus resistance. In the generation 28, that is in 2019, we fully implemented the genomic selection. And today, in 2020, we bought another strain, Aqua America. And today we are in G30 or generation 30 in GenoMar Supreme Tilapia, and then fully implemented genomic selection. So this has been the different phases. I'll briefly describe the different advancement in terms of 2 T's. First T is technology, and the second T is threats. And then I don't need to go through it in details because John and Marcela has described this process very beautifully. So initially there was mass selection, and then it started pedigree-based selection, that is family-based selection. And then another step was marker-assisted selection. Then now where we use very few SNPs or very few markers from the DNA. And then it was genomic selection where we use around 50 to 60,000 SNPs from the genome. And then whole genome sequencing, functional genomics, and as they said, and the gene editing. And the threats, more complex in tilapia, more complex selection to identify best animals has taken place. And if I have to describe it into the progression of order, I think this has been becoming more complex with more threats, from low average body weight to increase, and then the fillet yield, robustness, and different specific disease resistance. So how do we see it in the future? In the future, in terms of threats, I think it will be more complex selection to identify best animals, which are better in terms of production and quality threats, functional and fitness threats, and sustainability threats for Nile tilapia. And primarily, we assume that the production and quality threats will be around growth, fillet yield, feed conversion ratio, and meat quality. Similarly, the functional and fitness threats will be primarily focused on rusticity, livability, reproduction threats, deformities, and different specific disease resistances, like Aeromonas, Streptococcus, Franciscellosis, Flavobacterium, tilapia leg virus, and iridovirus. And then the sustainability threats. Probably it will be cost of production, sex control, lower emissions, resource efficiency, and temperature tolerance. So that's how we envision that will be in the future in terms of threats. I just described this in terms of threats and technology, how GenoMar Supreme Tilapia has gone it. So in technology, from the pedigree-based selection to in 2019, we started fully genomic selection in 2019-2020. And then for the threats, we went through growth from fillet, robustness, and specific disease resistance. And in our another strain, AquaBel strain, with more than 25 years of R&D and breeding work in Nile tilapia, again through different phases. We started the breeding program in 1994 for the AquaBel, and then in 2017, GenoMar Genetics bought the strain. So it has gone through mass and pedigree-based selection to the genomic selection. From 2020, we have started to implement genomic selection. And for the threats, currently we are selecting it for growth, fillet, threats, robustness, and specific disease resistance. So the two increase in the technology and increase in the threats are more complex, the more threats in the selective breeding. What does this mean to farmers? The first one, with the advancement of technologies, the advantage is the increase in the prediction accuracy. So here are 5 different threats: growth, fillet yield, Streptococcus columnaris, and Franciscellosis. So we can see that the different prediction accuracy, increase in the different prediction accuracy can be observed from family-based selection to genomic selection. And to the farmers, it means higher genetic gain means more better products or more sustainable products. And more traits in selection program. This means the genetic gain for all the desired traits, not only because as Marcela described that growth and the disease resistance for specific disease resistance, it was negatively correlated. So it might be various kinds of those types of negative correlations. So when we have more desired traits in a selective breeding program, we increase favorably for all the traits. And with more traits, we are able to introduce 2 different products, at least in the Asian market. where we have GenoMar Gen, that is Genomic Selection, has been applied since 2018 and available to farmers in 2021. And this provides large increase in accuracy of selection, leads to 20 to 30% improvements against the baseline annual rate of genetic gain for growth, fidelity, and robustness. Whereas we have GenoMar Strong, another product which has been selected for the disease resistance, especially streptococcosis. And as we know that streptococcosis is a devastating disease in tilapia aquaculture. And then we have been selecting for the breeding for strep resistance over 5 years. And this was the field trial where we did in the case farming in Malaysia, where we compared the streptococcosis resistant tilapia and non-selected tilapia. And we found that the streptococcosis resistant tilapia had the relative percentage survival of around 30% in version 1 of the product. The next version will be this version 1 of the product is already available in the market. And the version 2 of the product will be the genomic selected product. Briefly, we do publish lots of scientific literatures which can be found there, and then yes, for different disease resistance and the science behind what is being done in our breeding nucleus. And thank you for listening to me. If you have any questions, please do you can send an email even to me. Yes, thank you, Anna.
Thanks a lot, Rajesh. Very nice presentation. We go to the final presentation today. That's a presentation by Hans Halle-Knutzen. He's the Sales and Marketing Director of AquaGen. He has been having this position since 2019. He came from the same position in BioMar Norway, a position he had since 2013. Hans has a long and diversified career, such as experience from aquaculture working in Lerøy at Salmon seaside in mid-Norway for 5 years. His title is The Genetic Potential in Growth. Please, Hans.
Thank you for the introduction. Can you hear me and can you see my presentation?
Yes, looks fine.
Good. I have had some technical issues, so I'm glad that it works. Okay, thank you for the invitation. I will try to take you through the genetic potential in growth, or parts of it at least. The introduction, I think we skip. We are a little bit behind time, so I will try to speed up a little bit. Just first, a quick brief about AquaGen. Rajesh, he already has shown the different where parts of or our owners and AquaGen is part of every group. For AquaGen, we are a breeding company that develops, produces, and delivers genetic material to the global aquaculture farming industry. Actually, we have our 50-year anniversary this year. So it was 50 years since the first salmon was collected from 12 Norwegian rivers, and this was the beginning of our breeding program. So I will just take you through the genetic progress in 11 generations of salmon, and also try to say something around what you could expect through world-class breeding. So to start off, and balanced breeding, even though I'm going to talk about growth, balanced breeding is the fundament of our breeding program, ensuring optimal performance and responsible aquaculture. Resilient against all common pathogens and parasites in aquaculture, deep red fillet color and superior quality, robust fish that tolerates transport, handling, and treatment, and the last one, the good growth, and then shortening production time in both freshwater and seawater phases. And I would say the best possible growth. I'll there has been some excellent presentations that has in an excellent way described the basics of the breeding work, but just repetition is always good. It's the breeding is all about selecting the best parents, so the next generation will be better than the previous one. That's a simplification to a set. To have genetic progress, the genetic gain for a trait, the trait has to be heritable. There must be an observable difference between fish in the population, and this difference must partly be due to variation in the DNA. How much the DNA determines the variation can vary between 0 to 100%. As an example, you have the eye color in the humans that is almost 100% determined by the DNA. The genetic progress of trait are also decided by the intensity and accuracy of the selection. And I've been talking a lot about the tools also today, but again, when it comes to accuracy, it's about finding the right animals. All individuals are different, even siblings are different due to variations in their DNA. Some fish will grow better, others will be more resistant to different diseases, and others will have more red fillet color, as an example. The accuracy of selecting the right parents has improved significantly over the last years, and today we use state-of-the-art technology for finding the best individuals, the best parents based on their DNA information. Some traits are controlled by one or few genes and is easy to select by marker-assisted selection, like IPN resistance, also mentioned earlier, in Atlantic salmon, or breast cancer in humans. It's like finding Mount Everest. It's easy to see the tall mountain in the mountain chain, but most traits are controlled by many hundred of thousands of genes. And separately, the gene markers, or the SNPs, have a small effect, but combined together, they make a large effect. Heights in humans are traits like this, where height is very heritable and controlled by many markers, where the strongest gene marker only adds 1 to 2 millimeters. Using the mountain as an example, the small mountain tops may not be the high alone, but having them on top of each other would make a very high mountain. Growth in salmon is also controlled by thousands of genes, and genomic selection is an excellent tool for finding the biggest fishes for breeding and secure a good genetic gain of the trait. So growth has been a very important trait in AquaGen's breeding program for 50 years. And even without DNA information, we selected fish based on the families that grow best. Today, we have our 13th generation of Atlantic salmon in our broodstock. And a couple of years ago, our breeding manager wanted to demonstrate the genetic progress of growth in our population. To do so, he stripped eggs from females of the same generation. This was the 11th generation, and divided the eggs into different batches. Each batch of eggs was fertilized with sperm from males from different generations, all from wild salmon. This was the generation 0 then, and up to generation 11. After fertilization, the eggs were kept together in one tank. They were in a way mixed and bred in one net pen, keeping them from the same, keeping them in the same environment until harvest. So this would demonstrate the genetic contribution of growth, but only from the fathers. And if we look at the result, and you have from generation 0 in a way, and up to generation 11, we saw an increase of or the result, it demonstrates the genetic gain with a growth difference of 1.6 kilos. And this is only from the, remember, this is only from the male. So the growth potential, if you also accounted the female, would potentially be almost a double. And also, if just to show the potential, in each of these groups, there was a huge spread in growth performance, demonstrating that there are individual differences. There is a huge potential in each generation. And if you look at the circle here, and in generation 10 and 11, you see fish upwards to get, at the same time, you see fish almost 7 kilos. And this demonstrates the potential. And the average is again, well, to put another, fish that is twice the size as the average of the population. So if we look at the generation progress of growth in real life, call it, it's also appealing in the commercial freshwater production. I just wanted to show you these data from one of our customers here in Norway. With this is a site with a flow-through system, and they have operated this unit more or less with the same temperature regimes every year. And you see the smolt weight for a period of 5 years have increased. You see that on the left-hand scale. And as the numbers of days from start feeding to smolt transfer has been reduced. And production in freshwater facilities have often limitation with many bottlenecks, so it's not always easy to utilize the growth potential. But still, we get a good example of how smolt weight increases and how the number of production days decreases over years. If we look and see and look at the sea, if we tried to predict what we could expect going ahead in terms of increased growth, we see here that at the generation 0 or 1, we started between 600 and 700 days production days at sea, and we now have the 13th generation are looking close to 200 days at sea until 4 kilos. And where will this take us? It's not easy to predict, but still the curve is steep. But of course, in a certain time, it will flatten out in a way. And also just to mention the intervals, there are for salmon, and for salmon it's 3 years intervals. So today AquaGen is providing products adapted for the different production environments. And also, I think is a very interesting observation on the growth trait is that the growth trait seems to be a universal trait. The families that perform best growth-wise, they perform best independent in a way of the environment they live in. So if it's land-based or sea-based, it doesn't really matter as examples. So that was what I was going to present today. So thank you very much. And I leave it over to you, Anna.
Yeah, thanks a lot. Yes, so we have heard 4 very interesting presentations. We have got some questions in the Q&A. I would like to invite some of the speakers to answer a couple of them. I can start with John. If you could, for the whole audience here, say a little bit about the costs of these different breeding programs. Yes, I understand that there are many variables, but if you go from a more simple breeding program to a genomic selection program, how should you think about genotyping costs?
This is a good question. So, as you said, it's highly dependent on species, the genomic tools that are available, and demand. But I can give you an example for in Vaname, so white leg shrimp, which is an important aquaculture species. The cost is $9 or $10 to do genotyping for 200 SNPs, somewhere in that range, to do pedigree assignment or look at diversity. And then we were able to get enough people interested in the chip that we put together a 50K chip, and we can offer that for around $20 per sample. So you can get 50,000 SNPs for $20 and 200 SNPs for $10 with some variation there. But that's the general cost range. I think Vaname has a high use, so we were able to get good pricing there for a new species. Let's say you're working in Cereola, where there's less demand, it's an emerging species, then the costs are a little bit different, but customized solutions are still possible. So I'd say ballpark somewhere in that range.
Yeah, and if you go to higher density, what happens then to the price?
Yeah, so
like
that's,
if you would sequence.
Yeah, so that's a great question. For the work we're doing in rainbow trout, we've gotten to the point where we can do skim sequencing, so light sequencing, and there are great genomic resources and impute to there. And for there, we're looking at about the same cost as the commercially available chip. So they're equivalent. So you can either get 50,000 SNPs or we're getting like 2 to 3 million SNPs per individual. So, and then for new species, it's less sensitive to demand because there's no manufacturing. So again, if you take Cereola, probably it's a very small genome. It's dependent on genome size. So the cost there, it's probably cheaper to take that approach than to try to build a chip for something like that.
Yep. Good. Thank you. I also have another question to you. Where do you see that genome editing will go into the breeding program? Will you have like WISO genetic breeding programs in the lab, or will it be more connected to an existing breeding program?
I think it'll be just a tool in the breeding toolbox. So you'll still have the foundational family-based breeding program, or genomic selection will still continue. You'll just create a variant that you add to the program and then select for it. So I don't think it will replace, it won't replace breeding at all. It will only be a benefit. Otherwise, the structure of a breeding program, I don't think will change.
No. All right. Thank you.
And I'll point out just one last point. Even when you do an edit, or in the past work with transgenics, and you have this really amazing increase in performance, there's still variation, and you can still select even in the edited population.
Yeah.
So you can still make the 8 to 10% benefits per generation.
Yeah. Very good. Thank you, John. Our second speaker was Marcela. You got a question about EMS. If you have results on this disease, maybe you can say a little bit about this disease. It's a bacterial disease for the shrimp.
Yeah, EMS is a nightmare because, as I told the regulatory authorities in Colombia when I had to import the strain to do a challenge test, I said, you are going to read in the same paragraph a bacteria, a plasmid, and a toxin. It's a bacteria that carries a plasmid that produces a toxin that kills the shrimp. So it's a whole package of problems. We are doing challenge tests now for them, and we have had a good heritability around 0.3, and the plan is to start genomic selection for that. But first, as John said, we have to have a very good challenge test. The phenotype is the most important part before going to a genotype. If we don't have good information during the phenotyping, then your genotyping is just trash. You have to have a very good foundation on the phenotype. So we have a very good challenge test. With the pandemic, we had to stop a little bit because the university where we work was closed. So that's what has stopped us, but we are working on that this year too.
Yeah. Thank you. I have a question on this immune priming effect. what does it do to your genetic analysis? What do you think that you are looking at there? Is it epistatic effects or?
It seems to be epigenetics, actually. The papers, at least in insects, where it has been published, especially in bees, is that it's epigenetic effect, and it's transmitted mainly by the male. So what it seems that we are looking on immune priming is epigenetics, not genetics. We checked the effect on that, but it's difficult because it also confuses with the parent. But we did some experiments, for example, using actual survivors from the disease and the siblings, and we saw a very much increase in the survival of the progeny of the infected survival animals than on the others, on the siblings, for example. But it was confused. But it seems that immune priming is a phenomenon that we have to look at, especially in selection.
Yeah, that's very interesting and new, I think. There was, I don't see any new questions for you. So if Rajesh, there's one question here about just the tilapia farming. Have you seen that question? Yeah. It's if you have in which kind of water you have it, for example.
Yeah, I just answered it because generally tilapia is a freshwater species, so it's generally cultured in freshwater species, but with some crossing and every crossing, at least with Mozambicus, there are some strains which can be grown in the brackish water or seawater also.
Yes. Yeah, I think there are other tilapia species that are salt water. I have a question for you, Rajesh, on what is the biggest gap for the tilapia breeding? Because it's already, as you showed your graph, it's already increasing immense. But with such an increase, you also get more difficulties or problems are popping up. So what do you think is the biggest issue for your industry, and how can breeding react to them?
I think at least currently, I think diseases are the most challenges for all the aquaculture industries. And many diseases, new diseases are coming, and then again, it takes some time to do the selective breeding. And then, yes, I think that's the main challenge. And then at least in tilapia, fillet yield has been one of the challenges to improve the fillet yield. Yes, and we are doing lots of researches in novel phenotyping methodology to improve the fillet yield and these things. So I think these two threats, in terms of threats,
And is it difficult for you when you go out with your customers to actually convince your customers that breeding has an advantage? Because we saw here with the case of the salmon, it's a huge study done to actually show the genetic gains.
Well, tilapia, if we think about tilapia, tilapia is cultured in lots of different countries, in lots of different culture systems. And then it also depends on the customer base. In a country like Brazil, where they primarily the commercial product or the farmers grow the fillet. So they know the importance of the selectively bred individuals when they get the total output. Whereas in the table fish and those kind of farming practices, I think it's a little bit difficult to, for all the farmers to understand.
Yes. Good. Thank you. For Hans, I didn't really see any questions in the chat, I think, or are they coming any now? For example, here, what does selective breeding mean for fish welfare? I can give that to you, Hans.
Thank you. That was a well. Of course, that fish welfare is a hot topic in a way, at least in the industry, and I think it's has a huge potential on the genetical part, if you see it like that. We are constantly working on this, and I think hopefully we will also show new results going ahead. But definitely this together with other tools, we have to do our part to increase the fish welfare in today's fish farming.
Yeah. There was a question here on how long after initial starting the breeding program can you expect to see gains?
Sorry, how long?
How long after initial starting of the breeding program can you expect to see genetic gains? It's a very basic question.
Yeah, that's a little bit hard to answer, I think, depending on what you are aiming for. But as I said, for salmon, we have now a generation interval for on 3 years, and of course we are getting better and better tools, as also explained in the presentation. So I would say, and it's gonna we're gonna reduce the time between every time we get results in a way going ahead. But giving a specific answer, I think it's to that question.
I don't want to say anything. No.
It's hard.
Yeah. There was one in the. Sorry, I need to search here for the questions. They're jumping around a little bit. Here is one for you, Marcela. Almost every shrimp genetics company claims its genetics has a variety of benefits and advantages. Is there a commonly acceptable way to audit its claims? You're muted.
Yeah, everybody says that they have the best survivors and everything. I think that the best way to try this is to test them and to look the performance in the field. We can say everything we want with our animals in the lab, but the real answer is in the test. And for example, we are working hard with an SPR animal that is more robust and resistant, and we have had very good results in terms of survival in field tests and commercial tests. But we are all aiming, and most of the genetics are very good. Most programs are really good, and it's also how to adapt the animal to the conditions. So some programs will be much better for some conditions. For example, the SPR programs will be better for those commercial productions where biosecurity is not as strict, while the pure SPF growth lines will be very good for the really, really good biosecure conditions in control ponds. So I think that the genetics and the environment will interact. So it's time of finding the best program for the best conditions also. But it's the commercial test that will tell who's the best.
Yeah. Thank you. Rajesh, there is a new question here. Is there an alternative to use of methyl testosterone for monosex tilapia production?
Well, yes, there has been temperature treatment during the initial phases to change the females to males. There has been researches going there, but it has not been fully implemented in the commercial breeding programs. And there has been YY super males again. It has been practiced to generate the YY super males through the systematic mating to have all monosex tilapia, male tilapia.
Yeah. Thank you. There's a question here. Is it safe to eat hormone-treated tilapia? Or
Until
is it
now,
safe with breeding for consumers?
Yes, because the hormone treated are in the earlier stages of the life, not afterwards. So yes, and until now there has been no such researches done that shows the opposite effect in the health.
No. But also none that shows the other effect.
No.
Positive. Yeah.
No.
No studies at all. All right. Yeah. I'm asking Lucia, coming to an end of this webinar. We can go on. We have some more questions, but yeah.
Okay, so thank you very much.
Okay.
Jon.
Yeah.
Yeah. Thank you very much,
And,
Jon, Marcela, Rajesh, and Hans, and Anna for sharing your time today. And thanks everyone for joining in. If you want to stay up to date on the latest news on aquaculture hatcheries, I encourage you to subscribe to our publications on our website, hatcheryfm.com. See you next month for the third webinar in the series that will focus on feeds for hatcheries. This session will be held over 2 days on May 17 and 18 with major players in the feed industry. We hope to see you then again. Stay safe. Goodbye.
Thank you, everybody.
Bye, everybody.